Hello everyone, welcome back to my channel IGCSE Biology Help. In today's video, I'm going to be solving the February-March 2024 paper 6. This is the variant 2. Make sure to subscribe to my channel and like my videos so you can receive more content like this. Let's start solving. So question number 1 says, a student investigated the effect of glucose concentration on the rate of anaerobic respiration in yeast. So the glucose concentration is actually the independent variable, as I explained in my paper 6 videos that I did like a year ago. Anaerobic respiration in yeast breaks down glucose to form ethanol and carbon dioxide. Anaerobic respiration in yeast causes the blue dye, methylene blue, to become colorless. The time taken for the blue color to disappear can be used as a measure of the rate of anaerobic respiration in yeast. The student used this method. Label one test tube 0.0%, one test tube 0.5%, and one test tube 1%. Put 5 centimeter cubed of water into the test tube labeled 0%. Put 2.5 centimeter cubed of 1% glucose solution and 2.5 centimeter cubed of water into test tube labeled 0.5. Put 5 centimeter cubed of 1% of glucose solution into the test tube labeled 1. Stir the contents of the beaker containing the yeast suspension with the glass rod. Add 5 centimeter cubed of yeast suspension to each of the test tubes labeled 0, 5%, and 10%. So you're going to be adding the same amount of yeast in each of them. This is actually like something you have to be keeping constant, or else the experiment will not be valid. Put all 3 test tubes into water bath at 40 degrees. Start the stop clock and wait for 3 minutes. After 3 minutes, remove the test tubes from the water bath and place them in a test tube rack. Step 10. Use a pipette to add 1 drop of methylene blue dye to each of the test tubes. Carefully mix the contents of each test tube with a glass rod. Use a second pipette to slowly add a layer of oil to each of the test tubes. The layer of oil will float on top of the yeast suspension and methylene blue mixture as shown in figure 1.1. So, what is the use of the layer of oil? They have said over here that it is to maintain the anaerobic respiration conditions. So they may ask this question. Now, step 12 says, put the test tube back into the water bath and restart the stop clock. Step 13 says, measure the time taken for the blue color in each of the test tubes to disappear. Record the time taken in seconds for each test tube. The students stopped timing if the blue color had not disappeared after 10 minutes. They recorded this result as 600 in their table. The stop clocks from step 13 are shown in figure 1.2. Prepare a table to record the results of this investigation. So you have to also convert. Be sure to read the question. You have to convert to seconds. So over here there are in minutes. So the 0% glucose solution, of course it does not have glucose, so there is no color change, and they said they have stopped recording after 10 minutes if there is no color change. So there is no color change, and if you want to convert from minutes to seconds, you have to multiply the amount of minutes times 60, because we know in 1 minute there's 60 seconds. So this is going to give me 600 seconds. You're going to be doing a table just like this. Half of the table is going to be percentage concentration of glucose. This is actually the independent variable, and the time taken to disappear, of course, it is 600 seconds. I just converted it. Now for this one, the 0.5 concentration of glucose, it took 6 minutes and 2 seconds. So you're going to be converting the minutes first, which is 6 times 60, which is going to be equal 360, and make sure to add to the 2 seconds to the minutes that you have just converted. This is going to be 362. Now for the 1%, it is actually 2 minutes. It took less time, so you're going to be multiplying the 2 times 60, which is going to give you 120, and make sure to add the seconds as well. This is going to give you 132 seconds. You're going to be doing a table just like this one, and you're going to be getting your full marks. Next question. I state a conclusion for the results of this investigation. As the percentage of glucose increases, the rate of anaerobic respiration will increase and this will also decrease the time taken for the blue color to disappear, as we just saw over here in this table. Now, triple I says state the independent variable in this investigation. This is the concentration of glucose. As you all can remember or recall, in the beginning of the video, I said that anything that comes after the effect of, and I just said that information in my paper 6 videos as well, in planning an investigation. This is the independent variable because you're changing it. The thing that you change in your investigation, this is the independent variable. So our answer is going to be concentration of glucose. Now, IV says state one variable that was kept constant in this investigation. You can say temperature, you can say the time or the height of oil, or the volume of glucose or water. There's also a lot more answers, but this is like all that I can fit in here. So explain why it is important to stir the yeast suspension is in step 5 to evenly distribute the yeast cells. So stirring, as we all know, it is for evenly distributing the solution so that there's like all of the solution has equal amounts of, for example, the yeast in this example. So to evenly distribute. So anything that says why is it important to stir, it is always to evenly distribute the cells. B says one way to improve this investigation would be to use an increased number of different concentrations of glucose. Suggest 2 other ways to improve this investigation. So you can repeat the experiment 2 times and then take the average of the time taken for the color to change and exclude anomalous results. Anomalous results means results that are not fitting in the pattern. So you can make sure that your results up there was actually accurate by repeating it again and taking average or mean of the time taken. Also, you can use colorimeter for accurate color change. So sometimes there is errors or there's differences in the way that we judge colors, because of course all of us are different and our eyes will perceive color in a different way. So you can use colorimeter so that you can get accurate color changes and you can get a more accurate result. Double I says describe how you would make 5 centimeter cubed of 2.5% glucose solution using a 0.5 glucose solution and distilled water. So first of all, you're going to be this 5 centimeter cubed, you're going to be dividing it in 2 test tubes or 2 solutions. So the first one, you're going to be adding the 0.5% glucose, and then the second tube you're going to be adding distilled water to make it more, you're going to be making it less concentrated and more diluted, which means it will have more water. So if you want to decrease the amount of glucose, you have to add more water. So this is how you can dilute this solution. Triple I says describe the method you would use to test a solution for the presence of glucose. You can add Benedict reagent and heat in thermostatically controlled water bath at 80 degrees until the color changes from blue to brick red. So you can write also from red to, you can write from blue to orange, for example, or from blue to yellow, or any color that you know. But brick red will indicate that there is a high amount of glucose in this solution. But if you write any other color, then it can be correct. C says a student investigated the effect of temperature on the rate of respiration in yeast at 25 degrees and 35 degrees. In one experiment, the student measured the volume of carbon dioxide produced by the yeast every 5 minutes for a total of 30 minutes. Describe suitable apparatus that can be used to collect and measure the volume of carbon dioxide produced. Of course, you can use a gas syringe or inverted measuring cylinder. Any of these will be correct. The student did 3 experiments at each temperature. They used the results to calculate the mean volume of carbon dioxide produced. Part of the student's result table for the experiments at 35 degrees is shown at table 1.1. The student decided that the result of one of the one of the experiments shown in table 1.1 was anomalous. State what is meant by an anomalous result. I just said that in a previous question. Anomalous result is a result that doesn't fit the pattern. So, for example, over here we have 1.8 and then suddenly there is a 3.2. This is, of course, not fitting the pattern. Over here there is 2, and then there is the mean, which is 1.9. So what they did was actually exclude the result that is anomalous, because how are you getting 3.2 and the range is from 1 to 2, for example? So this is a result that doesn't fit the pattern. Describe how the student calculated the mean volume of carbon dioxide produced, shown in table 1.1. The student didn't include sample 2, only sample 1 and 3. So this is basically, you're going to get 1.8, for example, plus 2, and you're going to be dividing that over 2, so you get your mean value, or mean value over here, because you can't include this one because you're going to get a wrong result or a wrong value of your mean. So this is how you calculate your mean. The results of the whole investigation about the effect of temperature on the rate of respiration in yeast at 25 degrees and 35 are shown in table 1.2. IV, using the data in table 1.2, compare the mean volumes of carbon dioxide produced at 25 degrees and 35 degrees. So over here, we have the mean volumes. It's always higher in 35 degrees, as we see over here. And for example, at 5 minutes, the 25 degrees was at 0, and the 35 degrees one was at 0.1 already. And at 10 minutes, it was still 0 over here, and at 35 degrees, it is at 0.8. Now, the mean volume of carbon dioxide produced at 35 degrees is always higher, and we can say also at 25 minutes, the volume of carbon dioxide became constant, and at 35 degrees, in the first 10 minutes for 25 degrees, there was no volume of carbon dioxide produced. So you're going to basically describe the results as we do in paper 4. And we can see that at 25 degrees over here, at 25 minutes, it started to become constant over here for the 35 degrees, as we see over here. And the other one started increasing. It didn't become constant at 25 degrees. So this is how you answer this question. You're going to be describing the results, and you're going to be comparing both of them. Now, V says plot a line graph on the grid of the mean volume of carbon dioxide produced against time using all of the data in table 1.2. You will need to plot the data for each temperature as separate lines on your graph. Include a suitable key. So here's the key that I included. You can do anything, any shape, but you have to make different shapes that do not look like each other. So this is, for example, a triangle. It will indicate the 25 degrees, and the X will indicate 35. I drawn this graph. Of course, it doesn't look the best thing because I'm drawing on a tablet over here. If you draw on paper, it will be much more accurate than this, but this is just a demonstration of how it can look like for you. And also make sure to continue your lines. So over here, it should stop. This graph should stop here, and the other one should stop here, but you have to continue the graph. You have to continue like the line. So over here, I have labeled my axis. This is the x-axis. It is the time and minutes. I went on, this is 10 minutes, this is 20 minutes. You can, of course, distribute as you like, but this is the most efficient way of distributing. And I have plotted each of the points over here against time. So, of course, we can see that it became constant. For the 35 degrees, it became constant. Over here, it started to form like a straight line, and the other one was still increasing. Now, VI says estimate the time taken to produce 3 centimeter cubed of carbon dioxide at 35 degrees. Show on the graph how you obtained your estimate. So to answer this question, you have to do like this. So 3 centimeter cubed, right? So you're going to be drawing a line until you reach your graph that you drawn. So this is the 35 degrees one, and you're going to be drawing a line until you reach the time over here, which is the x-axis. And for me it turned out to be 24 over here. So it, of course, it's going to be different for each one of you guys, because of course it differs how your graph is. Not everyone has the same graph, so it's okay to get different numbers in this question. So it's 24 degrees. They will give you the mark even if it's a wrong answer, but you have to do this step, which is drawing the line until you reach the x-axis. So this is how you solve this question. D says carbon dioxide gas was bubbled through hydrogen carbonate indicator solution. The indicator was red before the gas was bubbled through. State the color change that would occur. It's going to be from red to yellow because they indicated over here that in the question that it was red before the gas was bubbled through. So we know that carbon dioxide is actually acidic, so it's going to be turning from red, which is neutral, to yellow, which is acidic. So you have to know the color changes over here. This is very important. Number 2, 2a, figure 2.1 shows epidermal cells from a red onion. And make sure to note the magnification. It's written over here. And let's see the question. Draw a large diagram of the cell labeled X in figure 2.1. Now, of course, my drawing looks terrible. This is because I'm drawing on a tablet, you guys, and it's very hard to like get them side to side. But this is the idea you should get. Like, you have to draw an outline. You don't shade. Never shade in your drawings. And don't draw like with the ruler or any tools. You have to do freehand. You have to use your hand, and even if it looks terrible, you're going to get your marks, and you're going to draw like an outline of the lines. Of course, you can draw it more accurate on your paper. You can draw, for example, you're going to be outlining this like this. I have made a separate video for this part of the exam. It's the drawings video, paper 6. So make sure to check it out if you are having trouble with this type of question. II line PQ on figure 2.1 represents the length of cell X. Measure the length of the line PQ on figure 2.1. So you're going to be measuring it using, this is the line over here. You're going to be measuring it on paper with your ruler, and you have to get from 0.226 until 0.232. This is the range accepted. So of course, this is because the paper differs for each one of us, and in the exam you should get one answer. So this is just for the printing differences. Calculate the actual length of cell X using the formula and your measurement. Magnification, the length of line PQ in figure 2.1, and then the actual length of the cell divided. Give your answer to 3 significant figures. Now, of course, I can't solve this question because I don't have a paper and a ruler, but we know that there is the triangle IM that I always explain in my videos. The I stands for image size, the actual size is the A, and the M is the magnification. So in this question, it is required for us to get the actual length. So you're going to be doing the image over magnification. The image size you may get over here. This is your image size, and the magnification, it is over 340. This is the magnification, and you're going to be getting your answer. B. Water moves into and out of the cells by osmosis. Plan an investigation to determine the effect of concentration of sodium chloride solution on osmosis in plant tissue. So first of all, you have to start off by saying your independent variables and your dependent variables, and then the independent variable is the 2 different concentrations of sodium chloride solution. So as I said, anything that comes after the word the effect of, it is the independent variable. So over here you're going to be saying that in your answer. And the dependent variable is the length or the mass of the plant tissue. So how you're going to be writing your method as well. Cutting cylinders, pieces of the potato. You're going to be cutting the potato into pieces and then putting the pieces into sodium chloride and measuring the length or the mass before and after. The like the submersion of the potato. Now the constant variables, you have to keep these things constant. This is the surface area and the initial length, or the initial mass, or the temperature. There's a lot of answers you can write down, and of course you can get a mark for saying you can repeat this experiment and taking the mean value or taking the average value. And you can also get another mark if you say, for example, you have to be using gloves or you have to be careful when you are cutting. And this is basically the end of our exam. Thank you all for watching. Make sure to subscribe and like so you can receive more content like this. And good luck with your exam.